So if you caught the virus, back in the early days of 2020, you probably made a very logical assumption.
Right. I mean, most people did.
Yeah, you figured, hey, I fought the real thing, right? My body went to war with the live virus, totally dismantled it, and learned every single trick it had.
It makes complete intuitive sense, honestly. We've been conditioned to think of natural infection as like the absolute most robust way to build immunity.
Exactly. You'd think surviving that actual physiological battlefield would grant you this ultimate, all-encompassing defense system. But what if that intuition is, well, fundamentally flawed?
Because the data is telling a very different story now.
Yeah, exactly. What if the sheer chaos of fighting a live systemic infection actually forces your immune system to panic? Like it cuts corners and churns out a defense grid that is practically useless against highly mutated variants like Omicron.
Which is a terrifying thought, but biologically fascinating.
It really is. So today we are unpacking a biological paradox in this deep dive. We're looking at a landmark 2026 study published in the journal Immunity by Werses and colleagues.
A really groundbreaking piece of literature.
Truly. And our mission for you, listening right now, is to map exactly how your body decides to build its tracking systems. We want to understand why a controlled mRNA vaccine exposure fundamentally alters the genetics of your immune cells, giving them the breadth to anticipate a virus's next disguise.
Well, a primary infection often leaves you biologically stagnant. What's fascinating here is the sheer microscopic scale of this analysis.
Yeah, it's not just a general blood test, right?
Not at all. This research isn't just taking blood samples and measuring floating antibodies in some general sense. The researchers conducted a massive multiplexed analysis of 4,797 individual human B cells.
That is just a staggering number of individual cells to look at.
Right. They didn't just want to know if you had a defense. They wanted to sequence the exact genetic blueprints of the microscopic factories producing that defense,
to see how their development differed based on how you were exposed in the first place. Okay, let's unpack this setup because we really need to understand what we are actually measuring before we get into the wild differences between vaccines and infections.
We definitely need to establish the baseline first.
Right, so if I'm looking at these 4,797 distinct cells, I need to understand what makes them special. We are talking about the B cell arsenal here.
Think of a B cell as a highly specialized immune factory, and its primary product is the antibody.
But it doesn't just shoot them out blindly.
No. Before it secretes those antibodies into your bloodstream, it wears a prototype on its surface. This is called the B cell receptor, or BCR.
BCR, okay.
Yeah, it is basically a membrane-bound version of the weapon.
So the BCR is the sensor. And, I don't want to use that old tired lock and key metaphor because it makes the immune system sound like a piece of static metal.
Right. It's way too rigid of a concept.
Exactly. It's so much more dynamic than that. Let's think of the B cell receptor as like a piece of facial recognition software.
Oh, I like that. That is a much more accurate way to visualize it.
Right. Because it scans.
Yes. The BCR is actively scanning the environment for an antigen, which is just a specific structural piece of the virus. If the BCR software recognizes the viral structure, it binds to it.
And that triggers the alarm.
Exactly, which activates the factory to start mass producing that specific antibody.
And this is where the Werses study gets incredibly ambitious. Because they didn't just test these 4,797 cellular software programs against the original Wuhan strain.
No, they went much further. They ran them through a multiplexed antigen panel.
A multiplexed panel, which means they tested every single cell against a whole progressive hierarchy of variants simultaneously.
Exactly simultaneously. They started with the ancestral Wuhan Hu1 strain. Then they tested against variants with slight mutations, like alpha and delta.
The ones we got used to hearing about in the news.
Right. And then finally, they introduced the highly evasive variants, the absolute masters of structural disguise like beta, gamma, and Omicron.
So they're throwing the hardest tests at them.
Yes. The goal was to measure breadth. And breadth is the holy grail of immunology. It asks, how many different disguises can your single B-cell software successfully recognize?
So if I'm tracking with you here, we are taking almost 5,000 individual immune factories. We're pulling the prototype sensors off their surfaces and running them through this gauntlet of increasingly disguised viral variants.
To see which ones are the absolute elite trackers, exactly.
And the way those trackers perform is deeply tied to how the immune system was first introduced to the virus. I mean, that's the core of it, isn't it?
It dictates everything. And this brings us to the core clinical observation of the paper.
Right, the big comparison.
Yeah, the researchers compared the B cells from individuals who received an mRNA vaccine against those who experienced a primary natural SARS-CoV-2 infection.
And the resulting cell profiles were just night and day.
Hmm.
So let's do a really quick recap for the listener so we don't lose anyone in the details here.
Good idea.
Okay, bullet 1. We are analyzing nearly 5,000 human B cells using a multiplex panel to test their breadth against multiple variants.
Bullet 2. BCRs, or B-cell receptors, are the unique tracking devices on these cells.
Right. And bullet 3. The whole goal is to see which cells can bind to the most variants, all the way from the Wuhan strain up to Omicron. So let's start with the mRNA vaccine response. What kind of factories did the vaccine build?
Well, vaccination preferentially stimulated a highly elite subset of cells called class-switched memory B cells, or CSMBCs.
Class-switched. Let's demystify that term real quick. It means the cell has fundamentally upgraded its machinery, right?
Exactly. When a B cell first activates, it produces a very large, somewhat clunky class of antibody called IgM.
Like a first draft.
Yeah. It's effective for a quick early response, but it's bulky. So class switching means the cell literally splices its own DNA to swap out the structural base of the antibody.
It edits its own DNA.
Yes, changing it to an IgG isotype. And IgG antibodies are smaller, incredibly precise, and can easily penetrate deep into tissues to hunt down the virus.
So the vaccine triggers these upgraded deep tissue hunting factories.
It does. Furthermore, within these class-switched cells, the researchers identified a very specific group they named Cluster 13.
Cluster 13, okay.
And Cluster 13 was characterized by the utilization of a specific heavy-chain gene called IGHV169.
IGHV169.
Paired with the IgG1 and IG3 isotypes. And the crucial finding here, these Cluster 13 CSMBCs demonstrated remarkably high antigen binding breadth.
Meaning they could recognize multiple variants. So the control vaccine exposure builds these highly adaptable Cluster 13 elite factories.
Yes, that's the vaccine path.
But here's where it gets really interesting. What happens during a natural live viral infection? You'd assume the body goes into overdrive and builds an even better version of these cells, right?
We would assume that, yeah. But the data diverges sharply from that expectation. Primary infection stimulated a completely different cellular profile.
A worse one.
Well, it overwhelmingly favored what immunologists call atypical memory B cells, or EMBCs.
Atypical.
Yeah.
I mean, that just doesn't sound like the elite squad.
It isn't. Genetically, these atypical cells relied on a totally different gene setup, primarily IGHV1/2, often pairing with a light chain gene called IGLV28.
So a completely different blueprint.
Exactly. And instead of the upgraded Ig isotype we saw with the vaccine, these atypical cells predominantly expressed IgA isotypes.
And the clinical result of that setup.
It was stark. This atypical profile resulted in significantly reduced binding breadth. Their facial recognition software was, well, rigid.
So if the virus changed its disguise even slightly...
These ATMBCs were blind to it, completely blind.
Wow, this is a phenomenal pivot point. Why? I mean, if you and I are trying to understand the logic of our own biology here, why would the natural path create a narrower, more rigid defense grid?
To understand the why, we really have to look at the environment in which these cells are born.
The actually conditions in the body.
Right. A primary viral infection is not a controlled learning environment. It is a state of physiological chaos. The virus is actively hijacking your cells, replicating uncontrollably, and destroying tissue.
Which sets off alarms everywhere.
Massive alarms. Your body responds with systemic inflammation, a cytokine storm. Your immune system is just screaming for immediate backup.
It's wartime mass production.
Precisely. The system doesn't have the luxury of time, so it hastily churns out these atypical memory B cells to neutralize the immediate threat.
Which works for the exact virus in front of them.
They work well enough against the specific strain causing the crisis today, yeah. But because they were rushed out the door, they lack the refinement to recognize the variants of tomorrow.
Okay, I have a mnemonic device for you listening, just to lock this in. Think VGG for the winning combo, vaccine, germinal center, which we'll get to, and IgG isotype, VGG.
That's a great way to remember it.
And then think IAA for the narrow path, infection, atypical cells, Ig isotype, IAA.
Perfect. So let's run through our second milestone summary. Bullet 1, mRNA vaccination triggers CSMBCs, specifically cluster 13, using the IGHV169 gene and IgG103, which leads to high variant breadth.
Bullet 2, primary infection triggers atypical B cells, using IGHV12 and IgA isotypes, leading to low breadth.
And bullet 3, The big takeaway here: natural infection does not automatically mean broader variant protection.
Which is huge. It really flips the script. But that stark difference forces us to ask the next critical question. How exactly does the body give those vaccine-driven CSMBCs their incredible breadth?
Right, where does that refinement actually happen?
Which takes us out of the bloodstream and into one of the most fascinating microscopic structures in the human body. We need to talk about the germinal center.
The germinal center is arguably a masterpiece of microanatomy. It is a temporary, highly specialized structure that forms within your lymph nodes or your spleen during an immune response.
So it's not always there.
No, it forms when needed. If your immune system is a military, the germinal center is its most elite, secretive research and development academy.
And the curriculum in this academy is a biological process called somatic hypermutation, or SHM. Now, I really want to spend some time on this.
It's one of the most important concepts.
Because when I first learned about SHM, it fundamentally changed how I view human biology. We always think of our DNA as this fixed, unchangeable blueprint.
But inside the germinal center, these B cells are intentionally scrambling their own genetic code.
It's just wild.
It is localized, hyper-accelerated evolution. The class-switched memory B cells, triggered by the vaccine, enter this germinal center.
And then what happens?
Once inside, an enzyme starts actively introducing random mutations into the specific DNA region that codes for their B cell receptor, their scanning software.
So it's basically running a machine learning algorithm in real time. The cell mutates its own code, changing the shape of its scanner slightly. Then it has to test that new scanner against a captured piece of the virus.
And the grading scale is brutal. If the mutation makes the scanner bind tighter to the virus, the B cell receives a survival signal and is allowed to multiply.
And if it makes it weaker?
If the mutation makes the binding weaker, the B cell is triggered to undergo apoptosis, programmed cell death.
So it's literally survival of the fittest, happening millions of times over, inside a structure the size of a pinhead. And this process isn't quick, is it?
Not at all. The data in the Wirtz paper shows a progressive increase in variant binding breadth over the course of 3 whole months.
3 months?
Yeah, the vaccine-derived CSMBCs sit in this germinal center for up to 90 days, undergoing round after round of mutation, testing, and refinement.
It's like an intense literary editing room. Somatic hypermutation is the editing process. The CSMBCs keep editing their draft for 3 months until it's a masterpiece that can read any variant.
That iterative editing is exactly what stretches their software to recognize the hidden, conserved structures that variants can't easily mutate.
But what about the atypical cells, the ones produced during the live infection? Do they go to this editing room?
They do not. The study found little to no evidence of somatic hypermutation in the atypical B cells. They just publish their rough draft and go home, essentially. They are biologically stagnant.
But wait, I have to challenge this point based on the paper. The researchers note that these atypical cells actually share clonal origins with the elite cells, right?
They do, yes. They share up to 90% nucleotide similarity.
Right, meaning an atypical cell and an elite cell can start from the exact same parent cell.
Yeah.
So if they share clonal origins, why does one get to go to the editing room and the other is completely shut out?
Well, if we connect this to the bigger picture, to that chaotic inflammatory environment we discussed earlier, the mechanism becomes clear.
Because the body is panicking.
Exactly. The researchers hypothesized that atypical B cells develop extrafollicularly, meaning outside of the germinal center follicle.
Because of the chaos.
Yes. The massive inflammatory signals of a live infection likely short-circuit the normal developmental pathways. The systemic panic pulls these B cell clones away from the germinal center.
And forces them to differentiate immediately in the extrafollicular space. So they bypass that 3-month editing process entirely.
Precisely. The controlled mRNA dose, however, doesn't trigger that systemic cytokine storm, allowing the clones to patiently enter the germinal center and undergo months of somatic hypermutation.
It's the difference between sending an engineer to a quiet laboratory for 3 months to design a universal sensor versus handing that same engineer a wrench in the middle of a hurricane and telling them to just fix the immediate leak.
That is a perfect analogy. And this brings us to the molecular level. We've talked about the editing process, but the editing can only do so much if the starting material is flawed.
Right, the blueprints themselves.
The researchers looked incredibly closely at the genetic blueprints, and they found that achieving breadth is heavily reliant on specific heavy and light chain gene pairings.
Okay, let's guide the listener through a recap before we look at those specific genes. Bullet 1, germinal centers are immune boot camps.
Bullet 2, Somatic hypermutation, or SHM, is the 3-month editing process that increases antibody breadth.
And bullet 3. Vaccine-driven CSMBCs undergo SHM, while infection-driven atypical B cells do not, leaving them stagnant. So moving into the genetics, you're saying that even if a cell gets into the academy, its success is mathematically constrained by its starting genes.
Absolutely. The B-cell receptor is made of protein chains, heavy chains, and light chains, which are encoded by specific genes.
So the genes have to match up correctly from the start.
Yes. The researchers found that baseline success against variants is dictated by strict, almost predetermined pairings. For example, BCRs that could only bind the original Wuhan strain heavily relied on a somatically mutated IgM with a bias toward lambda light chains.
So if you start with that, you're kind of stuck.
It's essentially a dead-end genetic blueprint when it comes to variants.
No matter how much you edit a bad blueprint, it's still not going to build a mansion.
Yeah.
But there are power couple blueprints, right? Genetic pairings that just inherently work better.
Yes, there are. For broader success, specific pairings are highly favored. The gene IGHV353 overwhelmingly pairs with IGKV19. This is a well-known public antibody pairing.
Why does that specific pairing work so well, though? Like, what is it actually doing physically?
It really comes down to structural flexibility and targeting. That specific heavy and light chain combination naturally folds into a molecular shape that binds incredibly well to the viral receptor binding domain.
The exact part the virus uses to enter our cells.
Exactly. It targets regions that the virus cannot easily mutate without destroying its own ability to infect.
It locks onto its Achilles heel.
Right. Similarly, the IGHV12 gene strongly favors pairing with IGLV214, 23, or 8. If your B cell randomly generates one of these power couple pairings initially and then enters the germinal center, it has a massive evolutionary head start.
It's biological matchmaking. But where is all this intense molecular editing actually taking place? Because when we talk about immunity, most of us just picture white blood cells floating around in our veins.
Right in the blood.
But this study went somewhere much more invasive to get their data. So what does this all mean? We always think of immunity in our blood, but is there another hidden laboratory?
There is, and it is one of the most striking aspects of the methodology. Blood only tells part of the story. It only shows you the cells that are currently out on patrol.
So where do they look to find the actual factories?
The researchers acquired lymph nodes and spleens from deceased organ donors.
The spleen. An organ most people only think about if it ruptures in an action movie.
True, but immunologically, the spleen is a titan. And what they found in the spleen tissue was remarkable.
What was it?
The B cells residing in the spleen showed even greater variant breadth than the B cells circulating in the blood.
Wow, so the blood is just the highway, but the spleen is acting as a hidden black box laboratory for our most elite antibodies.
Oh.
How did they prove that the mutations were actively happening there?
They measured the expression of an enzyme called AID, activation-induced cytidine deaminase.
That's the enzyme that literally cuts the DNA during somatic hypermutation, right?
Correct. AID is the chemical scissor that drives the mutation algorithm. The researchers found high active expressions of AID in the spleen tissue.
So the editing room is open for business.
Yes. This proves that ongoing long-term immune editing is happening deep in the spleen, continuously refining those CSMBCs long after the initial vaccine exposure has cleared from the system.
And they absolutely needed those elite spleen-trained editors to deal with the variants because the researchers charted a very clear hierarchy of viral evasion. The gauntlet gets incredibly tough at the end.
It really does. The binding results across the 4,797 cells showed a clear stepwise drop-off.
They started dropping like flies.
Yeah, most of the trained antibodies could handle the ancestral Wuhan strain. They held up decently against Alpha and Delta, which featured a few specific localized mutations like L442R.
But then came the heavy hitters.
Right, the structural floor fell out when they tested the highly evasive variants, Beta, Gamma, and especially Omicron.
Omicron BA.1.
Omicron's mutations were so extreme, so structurally distinct that it knocked out almost all binding across the board.
Even the atypical cells.
The atypical cells from natural infection were completely blind to it. The only cells that could successfully lock onto Omicron were the absolute most heavily edited CSMBCs.
The ones possessing the power couple genes.
Exactly, the ones that had spent the maximum 3 months undergoing AID-driven mutation in the spleen.
Omicron was essentially the final boss that only the 3-month spleen-trained veterans could even recognize. Okay, final subtopic recap here. Bullet 1. Specific heavy and light chain gene pairings. like IgHV353 and IgKV19, dictate baseline success.
Bullet 2. The spleen is a vital site for ongoing immune cell editing, which is that SHM process we talked about.
And bullet 3. Omicron is incredibly evasive, requiring a maximum 3-month edited CSMBCs to bind.
Which brings us to the ultimate synthesis of what this paper has uncovered. Immune breadth, your body's ability to anticipate and neutralize a mutating virus, is not just luck.
It's not random at all.
No, it isn't random. It is mathematically and biologically determined by a cascade of events.
First, the type of exposure, right? Vaccine versus infection.
Yes. A controlled mRNA vaccine prevents systemic panic, while a live infection triggers chaos. Second, that environment determines the resulting cell phenotype.
Whether you get the stagnant at MBCs or the elite CSMBCs.
Exactly. Third, it requires the luxury of time, up to 90 days spent in the germinal center boot camps running the somatic hypermutation algorithm. And finally, it relies on strict genetic pairings to serve as the perfect starting blueprint.
It is a stunningly complex mechanism, and understanding it fundamentally shifts how we view immunological memory. We've seen today that the controlled exposure of an mRNA vaccine actually gives your body the time and environment to send B cells to the germinal center boot camp.
Rather than panicking and churning out those atypical cells during a live infection.
Exactly. But this raises a question for you, the listener, to mull over. As our vaccine technology evolves, could we eventually design therapies that intentionally trigger specific genetic pairings like IgHV169?
It suggests a future of directed, programmable immunology.
Can we entirely engineer the editing room of our own spleens? Think about it. The next time you think about your immune history, don't picture a static brick wall. Picture a highly advanced, continuously running machine learning network.
Tirelessly editing its own genetic code in the hidden R&D forge of your spleen.
Thank you for joining us on this deep dive into the microscopic architecture of your own biology. We'd love to hear your aha moments, so be sure to share how understanding this changes the way you view your immune system. Until next time.